Assalamu alaikum dear students, welcome to your channel The Anatomy Canvas. In today's class of Embryology Foundation module, we are going to discuss with the mesodermal germ layers derivatives. So learning objective for the today's class are to explain the mesodermal derivatives, to explain the development of the somites, to describe development of intraembryonic coelom, to discuss the process of the vasculogenesis and angiogenesis and discuss their clinical correlates. So development of the mesoderm is that the process of the gastrulation. Initially the three germ layers are formed: the ectoderm, the mesoderm and the endoderm. So mesoderm is initially present in the form of the loosely woven connective tissue around the midline. On nearly day 17, this mesodermal tissue arranges itself into the form of the compact thickened plate of the tissue, and this is known as the periaxial mesoderm. At the lateral edges, this plate remains narrow and it forms the lateral plate mesoderm. Later on, the cavities appears, and when these cavities coalesce, they form the intraembryonic coelom, and they split the lateral plate mesoderm into two layers. The one which is continuous with the amnion is called the somatic layer of the lateral plate mesoderm, and one which is continuous with the yolk sac is forming the splanchnic layer of the lateral plate mesoderm. The part of the mesoderm which is present between the paraxial mesoderm and the lateral plate mesoderm is called the intermediate mesoderm. So mesoderm develops into three layers, that is the paraxial mesoderm, the intermediate mesoderm and the lateral plate mesoderm. This paraxial mesoderm, which is present around the neural tube and notochord, it organizes itself into the somites, and it gives rise to the axial skeleton, the axial muscles, the limb musculature and dermis. The intermediate mesoderm gives rise to the urogenital system, and lateral plate mesoderm will form the heart, that is its endocardium, the epicardium and the pericardium, the blood, the endothelium, wall of the gut and the wall of the respiratory tract. Their musculature is being formed by the mesoderm, and lining of the body cavities, that is the mesothelium. So these all are the derivatives of the three parts of the mesoderm, the paraxial, then the intermediate and the lateral plate. So now we will see how the somites are formed. So have a look at this diagram. This one is the ectoderm, and here the neural plate is forming, neural groove and neural folds are present, and here you can see the notochord. Around the notochord you can see this area is given the name of the paraxial mesoderm. So as notochord and neural tube forms, embryonic mesoderm on each side of them proliferate and form thick longitudinal column of the paraxial mesoderm. So this is the thick longitudinal column of the paraxial mesoderm. This column is continuous with the intermediate mesoderm on each side, and this intermediate mesoderm then thin out into thin layer of the lateral plate mesoderm. This lateral plate mesoderm is continuous with extraembryonic mesoderm, which covers the yolk sac and the amnion. So major subdivisions of the mesoderm are then, now look at this diagram. So this is the paraxial mesoderm. In the center you can see intermediate mesoderm and then the lateral plate mesoderm. Here you can see small cavities are present, and when these cavities coalesce they form a large cavity which is called the intraembryonic coelom or intraembryonic body cavity, and this intraembryonic coelom then split the lateral plate mesoderm into two. The one which is continuous with the amnion is called the somatopleuric layer, and one which is continuous with the yolk sac is called the splanchnopluric layer. So these are the three subdivisions of the mesoderm. In this diagram also you can see the somites which are forming the neural tube in its process of completion and fusion. So paraxial mesoderm differentiate and begin to divide into the cuboidal bodies called the somatomeres by beginning of the third week. These blocks of the mesoderm are located on each side of the developing neural tube. Nearly 42 to 44 4 pairs of the somites are present by end of the 5 week. In a triangular section, they form distinct surface elevation on the embryo. So please remember, first pair of the somite appears near day 20. That is in the 3rd week of development. Near day 20, first pair of the somite appears. From the 20th day onward, each day nearly 3 pairs are added, and total 42 to 44 pairs are present at the end of the 5th week. And out of these 42 to 44 pairs, there are usually 4 occipital, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 8 to 10 coccygeal pairs which are present. Out of all these pairs, usually last 5 and first occipital disappears and remaining form the vertebra. So these are your MCQs, that the first pair appear in the beginning of the 3rd week at the day 28. Then total number of the pair are 42 to 44 by the end of the 5 week, another MCQ. And third MCQ is that nearly 3 pairs are added each day, and by counting the number of the pairs even at this stage, we can estimate the age of the embryo. So this is another criteria to estimate the age of the embryo. In this diagram you can see, so this is the ectoderm and the neuroectoderm forming the neural groove and the neural folds. In the center you can see notochord. Here you can see paraxial mesoderm, which is forming the somites over here, and then the intermediate mesoderm and lateral plate mesoderm. And also remember, the section is taken over here. This is the level of this section. Now look at this diagram. Initially, this paraxial mesoderm forms somites, and these somitomeres are present in a form of a donut-shaped structure. They are arranged in the form of the concentric whorls around the center near the neural tube and the notochord. These somitomeres in the head region are given the name of the neuromeres. They are present with the segmentation of the neural tube, and they contribute to the mesenchyme in the head region. From the occipital region caudally, these somitomeres are called somite, and they will contribute to the formation of the body wall musculature. So these somitomeres later on then differentiate, and they form the 3 distinct patterns. So this part of the somite then show epithelial to mesenchymal transition, and the cells form the sclerotome, and this surrounds the neural tube and the notochord. The cells which are present at dorsomedial and ventrolateral edges of the somite, they will form the myotome component, and the remaining cells which are present over here, they will form the dermatome. So in this diagram you can see the sclerotome component, it has show from epithelial to mesenchymal transition, and it has migrated, and it will form the cartilage, the bone component, and the vertebra of that somite, while the myotome will slip beneath the dermatome, and it will form the muscle component, and dermatome will form the dermis of the skin and the body wall musculature. So these are the different components of the somite. So again I repeat that this component will become the sclerotome. This show a transition from epithelial to mesenchymal. Then this is the dorsomedial component and the ventrolateral component. These two parts become the myotome, and the remaining part form the dermatome. Collectively these two are called dermomyotome as well. So when myotomes slip beneath the dermatome, they will contribute to the muscle of that given segment of the somite, and this and the dermatome will form the dermis of that area. So first appear the somite first appear in the future occipital region, then they develop cephalocaudally. They give rise to axial skeletal and the associated musculature, also form the adjacent dermis of the skin, and the first pair of the somite appear near the end at approximately day 20. This is your MCQ, please remember. So first appear at a short distance caudal to the cranial end of the notochord. This is the location of the first pair of the somite, and subsequent appears in a craniocaudal or cephalocaudal sequence. So here you can see again the development of the somite. I have explained in the previous slide as well. This is a donut shaped structure forming the somite. This somite then differentiating into the dermatome, the myotome, and then the sclerotome component. During further stages of the somite development, you can see the cells forming the sclerotome has changed its character from epithelial to mesenchymal transition. They surround the developing neural tube and notochord, and they will form the cartilage, the tendons, and the bone component of that particular somite. The myotome will form the muscle component, and dermatome will form the dermis of that area. So here is an important concept given in your books about the lateral somatic frontier, and this concept shows you that the cells, some of the cells of the myotome of the somite crosses this area. This is the lateral somatic frontier, and they will form the abaxial muscle cells precursor, and while the cells which remain over here, they will form the premaxial muscle cells precursor. So this lateral somatic frontier is basically a border between each somite and the parietal layer of the lateral plate mesoderm, and it separates the two mesodermal domains. So cells which are crossing this area along with the lateral plate mesoderm will form the premaxial muscle precursors, and that the cells which remain over here will form the abaxial muscles, and they will form the muscles of the back. So this is an important concept which is given in your new editions. So this is the molecular regulation of the somite formation, and it is in the form of the human segmentation clock model. In an oscillating pattern, different genes are expressed. So in this diagram, you can see the human embryonic stem cells are taken, and they are activated to form the presomatic mesoderm to see the human segmentation clock model. So the notch proteins and notch and the WNT genes, these are activated in an oscillating pattern. Their concentrations are higher in the presomatic mesoderm, which is destined to form the somite, and when somites are formed, their concentration decreases. So there is an oscillating pattern in the notch and WNT protein. In addition to these protein, the border between the different somite, there is a different concentration of the retinoic acid and the fibroblast growth factor 8 and WNT protein as well. So retinoic acid is present in higher concentration cranially and lower concentration caudally, while FGF is present in low concentration cranially and higher concentration caudally. So there is the interplay of different genes and different proteins which are secreted and activated by these genes, which are regulated by these genes, and this in this way the human segmentation clock model is formed, and in an oscillating manner, the presomatic mesoderm then forms the somites. So molecular regulation of the somite differentiation, this is different from the previous slide, because in previous slide we were discussing the somite formation, and now we are discussing the somite differentiation, in which different factors are involved. So look at this diagram. This is the neural tube. This one is the ectoderm which is forming. This is notochord. This area is the somite which is differentiating into myotome. The dermatome, this area is again myotome, and this sclerotome portion, this is the lateral plate mesoderm, and different factors which are involved in this area are that the floor plate of the neural tube and these notochord secrete sonic hedgehog protein and noggin protein. These two proteins activate the ventromedial portion of the somite to form the sclerotome. So these two proteins basically activate the PAX1 gene, which is upregulated over here, and because of this PEX1 gene, this ventromedial portion of the somite then show from epithelial to mesenchymal transition, and these cells will become the sclerotome. So these cells will later on form the cartilage, the bone components of the vertebrae. Later on, another factor is released by the this area of the neural tube. This is the WNT and the neurotropic factor, and sonic hedgehog is also acting on this. These proteins activate the myogenic factor 5 and myogenic factor D. These proteins are activated over here in the ventromedial part of the somite and will form, this area will form the epiaxial or the abaxial muscle precursor, which will form the muscles of the back. Similarly, neurotropic factor, which is released from this part of the neural tube, will also activate the PEX3 gene, and as a result of this, the activation and expression of the PEX3 gene, this part of the somite will form the dermis. Another protein which is secreted by the surface ectoderm and the lateral plate mesoderm, these are the BMP4 and the WNT, which will activate again the myogenic factor D and myogenic factor F5 in the ventrolateral part of this somite, and which will activate this area, and this area will form again the myotome. So these are the different factors which are involved in molecular regulation of the somite differentiation. So this is the picture taken from your book, in which you can see this is the neural tube, and on each side of the neural tube you can see the somites which are forming. These are segmented in this area and unsegmented in this area. So this is the presomatic mesoderm, and here somite formation has taken place. This area is the cut edge of the ectoderm. Again, another diagram, this model is also present in your museum as well, so please have a look at this model carefully. This is the area where the nasal placodes are present, area where lens placode are present, area for formation of the midbrain. Here you can see the different somites which are forming over here. This is the area of the upper limb bud which is forming. This is the area where the mesonephric prominence is present, area where the lower limb bud is present. This is the caudal eminence. Look at this area. This is the left atrial prominence of the heart and left ventricular prominence of the heart, and these are the first, second, third and fourth pharyngeal arches. So in this embryo you can see a cylindrical embryo nearly a size of 4.5 millimeter. Embryo has now folded itself. This folding we will discuss in the next lecture as well. And so in this folded embryo, in which it is present in the form of a cylindrical body, you can see the development of the somites in addition to development of the mesonephric area, the heart which is developing, the pharyngeal arches, the lens and the nasal placodes, and also the upper limb and the lower limb buds which are present. So next is intraembryonic coelom. As I have already told you, the three parts of the mesoderm, the paraxial mesoderm, the intermediate mesoderm and the lateral plate mesoderm. So when cavities appear in the lateral plate mesoderm, these cavities will form the intraembryon. When they coalesce, they form the intraembryonic coelom, and they divide the lateral plate into somatic and the splanchnic layers. So appear as isolated coelomic spaces in lateral plate and the cardiogenic mesoderm. These spaces coalesce to form single horseshoe-shaped cavity. In this area you can see this is the horseshoe-shaped cavity which is present over here. This cavity is called the intraembryonic coelom. So parietal and the visceral layer of the mesoderm, the somatic mesoderm overlying embryonic ectoderm form the embryonic body wall or the somatopleure. So this is the somatopleure. In addition to amnion, these two collectively are called somatic pleure. Similarly, this is the chorion and it's a splanchnic layer of the extraembryonic mesoderm, and this is the splanchnic layer of the intraembryonic mesoderm. These collectively will form the splanchnopleury, and this splanchnopleury with underlying endoderm, they will form the splanchnopleury which will contribute to the embryonic gut. So, fate of the embryonic coelom now. During the second month, the intraembryonic coelom is then divided into three cavities. These are the pericardial cavity, which will surround the area where heart will be formed. Here you can see the pleuroperitoneal canals later on in this area. The pleural cavity is formed, and here you can see the peritoneal cavity. And at this stage, this peritoneal cavity is in wide communication with the extraembryonic coelomic spaces. So this is the fate of the intraembryonic coelom. This is a horseshoe-shaped cavity which will develop into the pericardial, pleural and peritoneal cavities. Now we will discuss the development of the cardiovascular system in the embryo. This cardiovascular system begins at third week of development when the needs of the embryo are not completely fulfilled by the process of the diffusion, and the embryonic tissue now need a well-developed system. So at this stage, the cardioprimordium of the cardiovascular system is laid down. So this development of the cardiovascular system is taken by the two processes. The first is the vasculogenesis and second is the angiogenesis, and it takes place in three areas. In the area of the yolk sac, where the process of the vasculogenesis and angiogenesis take place, in the area of the connecting stalk, and in the area of the chorion. Later on, a few days later, it also starts to develop in the area of the heart as well. So embryonic blood vessels begin to develop 2 days later from these. And this is because there is an urgent need for the blood vessels to bring nourishment and oxygen to embryo from mother, cause the early formation of the cardiovascular system. The primordial uteroplacental circulation develops during third week of intraembryonic life, and this is a very important MCQ as well. Until then, embryonic nutrition is obtained from internal blood by the process of vasculogenesis and angiogenesis. Formation of embryonic vascular process involves the two processes. The one is the vasculogenesis and other is the angiogenesis. So in vasculogenesis, the mesenchymal cell differentiate into the endothelial cells precursors called the angioblasts. These angioblasts aggregate to form isolated angiogenic cell clusters or the blood islands. Then the small cavities appear within these blood islands, and angioblasts flatten out to form the endothelial cells. So in this diagram you can see these are the blood islands. This is the wall of the yolk sac, and initially these are present in the form of the angioblast or angiogenic aggregates. When the cavities appear in this area, these are the cavities which are present. So cells surrounding these cavities, these will form the endothelial cells of the cavities, and cells which are remaining, they will form the precursors of the blood. So process of the vasculogenesis involves different factors, and these are first the mesoderm, which is destined to form the vasculogenesis. It is activated by the fibroblast growth factor, and it will form the hemangioblast. This hemangioblast is basically precursor for both the vasculogenesis and the angiogenesis. So endothelial cells arrange themselves around the cavities in the blood island to form the endothelium. So in this diagram you can see the cells derived from these hemangioblasts, they arrange themselves in a form of the to surround the cavity, and they will form the endothelial cells, and this process is regulated by the vascular endothelial growth factors, which regulate the endothelial cells formation. Then these endothelial line cavities soon fuses to form the endothelial channel, and process is called the vasculogenesis. So this initial process, the vasculogenesis, is formed initially first the mesodermal cells, which are destined to form the blood cells. They are activated by fibroblast growth factors. they will form the hemangioblast. Then the cells surrounding these cavities will form the endothelial cells by the activation of the vascular growth factors, and when these endothelial lined cavities joined each other, they will form the channels, and the process is called the vasculogenesis. And in process of the angiogenesis, the vessels sprouts into adjacent area by endothelial buddings, and they fuses with each other, and they will form the vessels fuses with each other, and they will form the angiogenesis. So basic process is vasculogenesis, and by the sprouting of the vessels from the initially formed vessels, the new vessels are formed, and then this process is called the angiogenesis. So development of the blood cells is that the blood cell develop from the endothelial cells of the vessels called hemangioblast, develop at the end of the third week on the yolk sac and allantois. Hematogenesis does not begin until fifth week of development. It occurs first in liver and later in spleen, bone marrow, and the lymph nodes. In the liver, it initially starts. The area is called the aorta gonad mesonephros region, because this is the region in which the aorta is developing, and also near this region the mesonephros is developing. So this region is given the name of the aorta gonad mesonephros. Cells from this area then populate the liver and later the spleen, and then the hematogenesis starts. And the fetal and the adult erythrocytes are derived from the different hematogenic progenitor cells called the hemangioblast. Mesenchymal cells surrounding the primordial endothelial cells differentiate into muscular and the connective tissue elements of the vessels. So primordial cardiovascular circulation, the heart and the vessels develop from the mesenchymal cell in the cardiogenic area. So here you can see this is the area, cardiogenic area, in which the mesenchymal cells are developing, and these are formed initially in the form of the two endothelial lined tubes, two heart tubes. When these two heart tubes fuses, they will form the heart tube. So paired longitudinal endothelial channels or endothelial heart tubes are present during third week, and these tubes fuses to form the heart tube. In this diagram you can see these tubes has the fusion has taken place in these tubes, and they have formed the heart. So primordial vascular circulation, basically tubular heart joined with the blood vessel in the embryo, the connecting stalk, the chorion, and yolk sac to form the primordial cardiovascular circulation. So in this diagram you can see this is the primordial heart which is forming, and it has joined the vessels which are present in the connecting, in the yolk sac stalk, in the connecting stalk, and in the body wall. So it has gained connection with all of the blood vessels in this area, and in this way the primordial cardiovascular system starts. So CVS is the first organ to reach a functional stage. This is again your MCQ. These are the clinical correlates with this area. These are basically capillary hemangiomas which are present, and these capillary hemangiomas may be focal or may be diffused. Focal capillary hemangiomas are present in nearly 10% of all live birth. They may be present at any part, but usually present in the area of the face. Facial lesion are most common, and in cases of the diffuse lesion, there may be ulceration, there may be blockage of the airway, and it can cause the serious complication. So these are the clinical correlates which are related with the formation of the vasculogenesis and angiogenesis, and these are basically are because of the insulin like growth factor 2, which is related in a increased levels and a higher levels, and leads to abnormal development of the blood vessels and vasculogenesis. So now we are discussing the last topic of the today's class, that is formation of the chorionic villus system. So first the primary villi are formed. These primary villi are formed the cytotrophoblast simply covered by the syncytiotrophoblast. When they acquire a mesodermal core, they will form the secondary villas. So primary chorionic villi become secondary as they acquire the mesenchymal core, and when there is a formation of the blood vessel in this core, they acquire the blood vessel, then then they are given the name of the tertiary villus. So before the end of the third week, capillaries develop in the secondary villus, and now it is called the tertiary villi. Then the further development of the chorionic villus takes place, that the cytotrophoblastic extension from these stem villi join to form the cytotrophoblastic shell that anchors the chorionic sac to the endometrium. So here in this diagram you can see these yellow color cells. These are the cytotrophoblastic cells. They migrate from the area of the villi and they come to lie at the junction point of the endometrium with the trophoblastic tissue, and here they form the cytotrophoblastic shell, and this cytotrophoblastic shell then help to anchor the chorionic sac to the endometrium. So the rapid development of chorionic villi during the third week greatly increases the surface area of the chorion. This causes exchange of the oxygen and the nutrients between the maternal and the embryonic circulation. So this is the revision slide of the today's lecture, the postnatal derivative of the embryonic mesoderm. These embryonic mesoderm differentiate into intermediate, lateral plate and paraxial. Paraxial mesoderm will form the dermis of the skin, the muscles both axial and limb, and the skeleton. And the lateral plate mesoderm then differentiate into somatic mesoderm and the splanchnic. Somatic mesoderm will form the skeleton, that is the limb, not the axial, and the parietal forming the pleura, pericardium and the peritoneum. Similarly, the splanchnic mesoderm then differentiate into visceral pleura, the mesenteries, the epicardium, the visceral blood vessels, endothelium. Similarly, myocardium, the respiratory tract, and the gut for musculature. Similarly, intermediate mesoderm will form the urogenital system, in which kidney and ureter are formed, but remember urinary bladder is not formed from the intermediate mesoderm. See, this is again your another MCQ. The adrenal cortex, the gonads, they are formed by intermediate mesoderm but not the gametes, because gametes are formed by the primordial germ cells which migrate from the area of the yolk sac. Then the vagina, the uterus and the uterine tube, they are all formed from the intermediate mesoderm. So these are the postnatal derivatives of embryonic mesoderm and its three layers, which are forming different tissues and organs in the human. So these are the references: the Langman Medical Embryology and Developing Human by Keith L. Moore. I thank you all, dear students, for your attention for today's class, and Allah Hafiz till the next class.